This is the Pharmaverse Podcast. I'm your host, Michael Pietrack, engaging leaders from the pharma universe. Hello and welcome back to the Pharmaverse Podcast. I'm your host, Michael Pietrack. And if you're a leader or an aspiring leader in the pharmaceutical industry, well, then you are in the right place because we interview the best, the brightest stars from all the different corners of the pharma universe. And today's guest would certainly fit that description. We're joined by Krieg. He's the founder and CEO of Zola Therapeutics. It's a biotechnology company focused on developing a suite of immune system activators. Art is a physician scientist. He's a renowned biotech entrepreneur and a student of leadership, and he's here to share his insights. So Krieg, welcome to the Pharmaverse.
Thanks very much, Michael. It's a pleasure to join you today.
Tell us a little bit about what you're developing at Zola.
So at Zola, we're trying to activate the immune system to treat cancer. And this is not a new idea. I mean, people have been trying to do that for a long time. We're doing it in a different way than people have done it before. Specifically, we're trying to mimic the signals that a virus gives to the immune system that let immune cells know that there's a viral infection. And this is because to fight a viral infection, the immune system mounts a type of immune response with CD8 positive T cells. These are the assassins of the immune system that evolved to kill viral infected cells. And we think that we can activate them in patients who have cancer to kill the tumor by mimicking the minimal signals of a virus. And those minimal signals are viral RNA and viral DNA. That's how the immune cells have evolved to detect viral infection.
That's so fascinating. And, for someone who's a non-scientist, it's like, why doesn't the body go and fight cancer off? Can you explain that to if there's a layman listening?
Yeah, no, absolutely. The reason is that tumors mimic damaged tissue. They're very good at this. They evolve for that purpose. A friend of mine, Michael Lotz, coined the term that tumors are addicted to death. What they do is in our bodies, they give off signals of damaged or stressed or dying cells. Immune cells come into this tissue and they're looking for infection. If they detect an infection, they will eradicate it and destroy the abnormal cells, but they don't detect that. They detect stressed cells. And immune cells come into traumatic tissue injuries all the time, and they play a role in healing damaged tissue. So they make a wound response, and that brings in new blood vessels, nutrients for the tumor. So ironically, they actually help the tumor grow. Now, once in a long while, immune cells can tell that a tumor is abnormal, and this is called immune surveillance, and sometimes they eradicate those tumors. But it doesn't happen as often as we wish it would.
So at Zola, what are you doing that is different? Maybe a delivery system or something like that?
What we're doing that's different is we are combining several signals together that have never been combined together before. And these signals are a mimic of viral RNA that activates two immune receptors called TLR7 and TLR8. The second thing we're doing, you talked about delivery, RNA and DNA are not very stable. They get degraded quickly. So we're delivering these using a lipid nanoparticle. It's a similar technology that's been used for mRNA vaccines. It's a standard way to deliver nucleic acid therapeutics. And there's a whole lot of different types of lipid nanoparticles that you can use. We're using one that causes the least possible inflammation because we think inflammation actually can help tumors grow, and we want to avoid that.
Well, how have the results been so far in early development?
So that's actually a really interesting question to address because the usual approach when you're trying to develop a cancer immunotherapy to test the results, you go into mouse tumor models. And this it has failed repeatedly in developing human therapeutics. Basically, anything that you do to activate the immune system will cure tumors in mice, and then they almost always fail in humans. So the approach that we've taken in preclinical development is to start with human immune cells. And then you have the question of what is the assay that you're looking for? I talked about inflammation. We don't want inflammation. The bad type of inflammation is cytokines like IL-6, TNF-alpha. The good kind of inflammation that we want is interferon alpha. So for our preclinical studies, we screen human immune cells to look for the highest level of interferon alpha that we can get. So we do human immune cells. We start with normal immune cells, just from normal donors, and we optimize for getting the highest interferon alpha that we can. Then we take immune cells from human tumors, because if we're going to overcome the immune suppressive environment in a tumor, then we have to be able to induce interferon alpha in those tumor immune cells. Once we have accomplished that, which we've done, then we go into monkeys. And we actually did monkey studies before we did mouse studies, because the monkeys mimic, they're much closer to the human immune system. And in the monkeys, we looked for, can we induce interferon alpha in the monkeys? At the same time, or following that, then we did finally go into mouse tumor models too, as well as mouse safety studies. And we've seen good results in all of those models, so really exceeding our expectations. The level of interferon alpha that we want is over 50 times higher than anything that we had achieved in the past using standard immune activators. So I'm super pumped about that.
That's so fascinating, Art. Well, we're all rooting for you. What's the next big milestone for Zola?
Well, we need to get this into humans. There's a lot more work we could do. So far, we have data in just, 2 mouse tumor models. The next model that we need to show activity in is humans. What we need, though, is funding. And, in today's funding environment, that is a challenge. So that's really the next big step for Zola, is raising the funding to finish our GMP manufacturing, do the IND enabling studies, and get this drug into humans, hopefully next year.
Again, we're all rooting for you, but I want to dive into your story because it's such a cool story. So before founding Zola, you were the co-founder of Checkmate Pharmaceuticals, which was acquired by Regeneron. So what led you to want to start another company after such a big sellout?
Well, I didn't want to at first. After Regeneron acquired Checkmate, I was really, happy to transfer this program that we had there to Regeneron. And then I went on vacation, and I was kind of going to retire and just consult for companies. But I thought more and more about what we were doing at Checkmate. I mean, this one guy who was in our trials at Checkmate, he lives in upstate New York, and he had melanoma, and he was treated with what was available. And then his doctor basically said, your tumor's progressing and you've got 6 months to get your affairs in order. Or you can drive 4 or 5 hours each way to Boston every week for 7 weeks to take part in this new clinical trial that they're doing there. And he chose to do that. And it's patients like that who help us learn what works and what doesn't work. He had a great response to our treatment. Thank heavens. Many patients didn't. And I want to build on what we've learned to bring good responses to more patients. I think we can do that. Science has progressed so far, but there's still we haven't put all the pieces together. And I kind of feel like over the last 30 years that I've been working on this, I've put some of these pieces together, and I just have to get this the rest of the way.
Well, let's talk about leadership now, because you said something in the pre-show meeting that was just really profound. You said, my philosophy is that there's always a path forward. My job as the leader is to find it. Now, how has that philosophy been a common thread throughout your career?
Well, in my first company, 𝐥𝐸, we were very focused on developing this new way to activate the immune system using mimics of viral DNA that we had at that time. And the path was kind of a branching path, because we looked at this technology and we said, we could develop this for vaccines. We could develop it to treat infectious diseases. We could develop it to treat asthma and allergies, and we could develop it to treat cancer. And we kind of went through the exercise of how are we going to do all those paths as one small company, and we decided to partner some of those. We ended up developing the cancer path ourselves, and there were some successes there. There are 2 approved vaccines that use that form of CpG DNA, it's called, as an adjuvant for the vaccines, but the cancer trials failed. And At the time, I thought there must be a pathway forward here, but this is now in the mid-2000s, and Pfizer acquired our company in 2008, and Pfizer was pretty actively involved in cancer immunotherapy then, but there were a number of failures, and by 2011, Pfizer made the decision to exit cancer immunotherapy. At that time, I talked to an investor about the space, and he said that the only way to make money in cancer immunotherapy is to short every company. And, he was right, because that was a winning strategy. So at that time, I did not see a pathway forward for cancer immunotherapy. A few years later, in 2014, the checkpoint inhibitors, the PD-1 checkpoint inhibitors were approved. And now things changed. And so that was an opportunity then to come back to this path with a new form of the CpG DNA, because we had realized that the original form we were using wasn't inducing that much of the interferon alpha that I mentioned is necessary for driving CD8 T cells. So for cancer immunotherapy, we have a new form of DNA then to drive more interferon alpha. That was the basis for Checkmate to get that funded. At Checkmate, we saw that pathway fairly clearly for how we could develop this. When we ran out of cash and the markets were down, that pathway went through Regeneron. Now at Zola, our pathway is to take this RNA DNA forward into the clinic. As a leader, I feel that my job for our team is to work together with the team to try to find that pathway, generate data that will support this. When we have investor discussions and the investors ask questions, come up with data to support those questions. We all in Zola believe this is going to work. We believe in the science. It's bad timing right now, but yeah, there is a pathway forward. And we're reaching out to academic collaborators too, to help us on this as well, generating data in some of their models to help support this.
I think sometimes the mistake that a leader makes is they get married to an outcome, and it keeps them from finding the path forward because they're like trying to jam it into this one path, and that might not be the path forward.
Yeah.
And so
that's such an important point. I remember talking when I was first getting into biotech, talking to some more experienced people, and they gave me multiple examples of companies that started on one path and realized early on that that just wasn't going to work, and they pivoted and found a different pathway forward. And I think that that's a really important point. Sometimes the pathway isn't to just keep hammering away at what you were trying to do. The bigger picture is if you want to develop new therapeutics, maybe there's a different therapeutic approach that's going to work better.
This episode is brought to you by 𝐲𝐻 International, the leader in pharma and biotech recruiting. When pharma executives need to make critical hires, they go to the experts at 𝐲𝐻, pharma's one-source staffing solution. Visit kbic.com. Now back to the episode. Now, you've had this amazing career. Tell us about a time where you had to face like a really big leadership challenge.
Well, there have been a lot of those, unfortunately, because of the failures that we've had along the way. And I think something that really helped me in my first company, I was not CEO, I was the CSO. And our CEO was a serial entrepreneur, Bob Bratzler. He had started multiple companies. And when we had the phase 3 clinical trial failures at my first company, Coley Pharmaceutical Group, I just remember feeling that I'd been kicked in the gut. But I just remember Bob was very calm and very matter of fact. And we just sat down as a group and talked through this, what are we going to do? How much money do we have? What can we do to make that money last? And that was a model for me of how Bob handled that situation, so that when I had situations like this later, that I thought back on that and I realized, first of all, you always try to be prepared for bad news and have a plan B. But when that comes up, you really need to just help your team work through this and find that new pathway forward that may not be what you thought it was going to be initially.
Tell us about how you were when you were starting off as a leader compared to today.
Oh, I was pretty hopeless. I was an academic physician scientist, and I did a postdoc at NIH and then started in academia. And no one ever told me how to be a leader. I don't remember having any leadership training. And all of a sudden, I'm in charge of a lab, and I have some people in the lab working. And I think one of the times I remember was when one of my senior lab techs quit because of a dispute with a different person in the lab that I just had not been managing. I was just sort of ignoring that, and I kind of knew it was going on, but I just ignored it. And I realized, I have to do a better job here managing these things. I can't just ignore them, because it's my job as a lab head to create an environment where people don't feel they have to quit because of disputes with other people. And I tried to read management books, and it's awfully hard. You read these books and it all kinds of makes sense, but then you try to put it into practice in the real world. But I realized I had to improve as a leader, and I actively sought out advice from other people, and I got executive coaches as well. Once I was in biotech, that's something I encourage every leader to take advantage of. If you have the opportunity to work with an executive coach, I just learned so much from my coaches over the years, and I'm still a work in progress, but you learn from your mistakes, I think, too. And one other point I'll make too, Michael, is everyone has strengths and weaknesses. And some people are really good at managing other people, and it comes naturally to them. You just have to be honest with yourself if it doesn't come naturally to you, and try to accommodate for that by the team of people that you pick and that you work with to make sure that they compensate for your weaknesses.
That's a really good tip. And any other advice that you would give to maybe a leader that is early on in their career? You talked about, getting a coach. You talked about, playing to your strengths. Anything else?
Yeah. For people early in their leadership path, when you're in a biotech environment especially, you really need to focus not just on leading your own group, but on your role in the team. And this is a big difference from academia that I've talked about with academic people who are making that transition into biotech. Academics, you succeed based on your publications and your impact factor. In a biotech environment, you succeed based on the whole team. And this is something I had some real heart-to-heart discussions with some of my scientists and some companies in the past, where they just were not getting along with other directors of other groups. And I insisted that one person work with a coach. She ended up leaving because she just couldn't work with other groups in a collaborative, cooperative way. A number of scientists who were great individual contributors, I put them on a plan where I said, you are going to be evaluated at the end of every year, not just by me, but by your peers in the other groups that you're supposed to interact with. And you will be evaluating them too for how well you are working as a team to help your scientists work together, share information, and get things done. And I think it's helped.
All kinds of different things shape leaders. Sometimes it's their childhood. Sometimes it's the pressure that was put on them. Other times it's just something that they do. And, getting to know you a little bit, it seems like you like to do these tests of fitness, like running marathons, biking, and that sort of thing. How has that shaped you as a leader?
I think running a biotech company, growing a biotech company, it requires a sustained effort over years. And you have to stay up, you have to stay focused, you have to be sharp, you have to be positive, upbeat. What I like about trail running, long distances, ultra marathons, it really trains your mind to stay focused over a prolonged period of time when you're tired and you're hungry and you're thirsty. And I think there's something there that carries over into the work environment too. I can't really define it, but I find a lot of times I'll go out for a really long run, and I'll have some problem that's been nagging me, and I come back from it and I take a break and have a snack or something, and then I see a solution that I hadn't thought of before. And I like to think that my brain, my subconscious, is kind of working on these things while I'm distracting my body with this physically demanding activity. I don't know. I find it fun, and I find biotech fun too.
Yeah, but it does it's very analogous because it is a marathon, and you got to bring it every second. And, it does take mental toughness,
It
so
does.
I see the analogy there. Last question. I ask every guest this because a lot of our listeners are aspiring to become leaders. They haven't had the opportunity yet. What advice would you give to those ones that are trying to maybe position themselves to move into a leadership position?
Yeah. I think I would go back to the team player bit before. Try to make other people look good. Try to help other people succeed. Some people think that they're going to get into a leadership position by making their boss look bad, and that their boss will leave and they'll be promoted into that position. I think that's a really bad strategy, because if your boss does get fired, you're probably going to get fired too, because they're just going to do a clean sweep. One other point I'll make too is, don't make the mistake of being silent when decisions are being made that you might disagree with. Sometimes people think, well, I'm at a junior level, I better not speak up. But if you know something that people haven't thought of and they're discussing a problem, a challenge, you can speak up. You can share your perspective on this situation, and maybe you have something people haven't thought of before. A leader speaks up not just when they're asked to speak up, but when they think of something that can help the group make a better decision as a whole.
That's a good point. And it's same is true when you are the leader. You don't want to be the emperor with no clothes and no one will tell you, something.
Yeah.
If there's a blind spot, call it out. Let us know.
Absolutely. Absolutely. You want to encourage dissent. You want to be the dissenter, and you want to encourage other people to be the dissenter too.
Fascinating stuff. Art, unfortunately, that's all the time that we have for this episode. But I have to say, on behalf of everyone in the Pharmaverse, thank you so much for your time and your valuable insights.
Thank you, Michael. It's a pleasure. I look forward to keeping in touch.
Absolutely. Well, that concludes another episode of the Pharmaverse Podcast. We have many more exceptional leaders lined up for you, so subscribe to the channel so you don't miss any insights. But for now, that's a wrap. See you next time on the Pharmaverse. Thanks for joining us on the Pharmaverse Podcast. Make sure you subscribe below so you don't miss any leadership insights. Like and comment. I would love to hear from you. We'll see you on the next episode of Pharmaverse.